what single cell RNA sequencing is it looks at the gene expression pattern of each and every cell in the sample. So if you think about, if you were looking at the previous technologies homogenized, so they were like a blender. So if you were looking at blueberries and strawberries, you would see a purple smoothie. Okay, but this new technology lets us see what's going on in each of those cells or each of those berries. So we don't have to homogenize and mix everything together. So we can understand what's going on in the cancer cells, and we can understand what's going on in the immune cells. So when we looked at our problem, our first question is, what kind of cell was mediating the response of the Merkel cell cancer? What made the cancer shrink? And we were able to see that there was an activated T cell population that was causing the cancer to shrink, and that it was the T cells that we had put into the patient that were making the cancer shrink. And so we said, okay, we know what mediated this response to the immunotherapy, but now how did the cancer get around it? And when we did the single cell RNA sequencing of the tumor biopsies, we were able to see that the tumor cells, and just the tumor cells, were hiding from the immune cells by putting down a specific gene that was necessary to show the antigen to the T cells. And so this new technology was able to give us an answer for how the cancer cells were getting around the immune system. And it was also able to suggest to us ways to make the immunotherapy better by putting in T cells that recognize more than one specificity. So more than one type of T cells at the same time might be associated with better outcomes in the future. And we're working to develop protocols now where we're going to do combinations of T cells moving forward.